We're here today to celebrate the 75th anniversary of the Avery-MacLeod-McCarty publication describing for the first time that DNA was a genetic substance. We will focus here on the contribution of Macklin McCarty, or Mac. Mac McCarty was a gentleman scientist. In the 43 years that I worked with him, I never had the impression that he was unhappy, that he and his colleagues never received the Nobel Prize for the finding of the century, that DNA was a genetic substance. He was more frustrated than angry. After their original publication in 1944, skeptics, some here at the university, soon claimed that it was likely impurities in their preparation that was the active substance. Being the biochemist of the trio, McCarty set out to prove it was DNA alone that was the transforming principle. To accomplish this, he purified DNase for the first time ever to specifically degrade the DNA in their preparation, showing that only this treatment removed the transforming activity. Still, it was argued at the time that the protein were highly complex molecules and thus fit the requirement to be the genetic material. There was so much controversy over their finding that years had passed before the concept was finally accepted, based on results of non-skeptics like Watson and Crick and Hershey and Chase in the 50s. In 1955, Avery had passed away, eliminating the possibility of the coveted award. The main organism under study in the Avery Laboratory of Acute Respiratory Disease was Streptococcus pneumoniae, the perfect organism to identify the transforming material. This is the one of few bacteria that have the capacity to take up free DNA into the cell and use it for their own purpose. Thus, they had a reliable system to identify the transforming substance. Had they been working on Streptococcus pyogenes or Staphylococcus aureus, for example, the experiment would not have worked, and it could have taken decades until the right experiments were performed at the right time in perhaps some other laboratory. Mac was a physician scientist of great distinction. For several decades post-1944, he led the groundbreaking work that established the cell architecture of the Streptococcus and the nature and location of its important antigens. By the mid-1950s, Mac began a series of elegant experiments to understand the nature of the cell wall and its relationship to the observed serological response to this organism. He was an exceedingly careful scientist and did not believe in publishing a series of short papers, each with small messages, but would wait until the whole story could be told in a larger, more complete work. Mac was an outstanding mentor, a model of professional integrity, and a statesman for science. He was a quintessential physician scientist. He did not deviate from his goal of applying his scientific expertise to patient-oriented research. Medicine and science has benefited from his contributions. I would now like to introduce Geoff Montgomery, a science writer who worked on special projects for President Torsten Wiesel in 1992 and 1996. He helped organize the 50th anniversary of the celebration of the same Avery-MacLeod-McCarty paper, and he conducted the 1998 video interview with Mac that you'll see here today. And now you'll get a really true sense of who Mac really was. Thank you.
We're not going to see the whole interview, which is 2 hours, but I did pay a visit to my friend Marge McCarty on December 14th, just to show her we had this video digitized, we're going to be having this event, and we sat down on the couch and I thought we'd watch 20 or 30 minutes of it. We watched 2 hours straight as the light faded outside the window of her lovely apartment. And it's a shame that I only am going to be able to show short extracts of this video, and I kept on putting things in and taking things out. And we're very committed, I've talked to Olga and other people here at the university, of getting this video out in a narrative form so the world can see it, because this is an undertold story. And it's kind of a difficult story because there's so many dimensions to it. And if you'll permit me a slight, perhaps self-indulgent personal anecdote that kind of brought it into focus for me. and it will lead to my first slide, and then we'll go to the first extract. I think after one of my meetings with Olga, and we were talking about the event, and I get very excited, as people who know me can say. I went to dinner with my 82-year-old mother, and I started talking. Remember this, event that I did? Was working on the 50th anniversary, and Mac-McCarty, and I did this interview, and they discovered the genetic secret of life, DNA. Genes are made of DNA, and it came out of an ammonia lab, and I'm going on, and she interrupts me. Speaking of that, I just had my pneumonia shot last week, and the next day, my arm was feeling pretty sore. And the day after that, I could feel a little bit, but by the third day, it felt fine. And I said, Mom, I'm talking about the genetic secret of life, one of the great discoveries in the history of civilization, and you're telling me, oi, my arm ached for a day after I had my pneumonia shot? I mean, let's have a sense of perspective here. But in the days following, I actually thought, and it's difficult to say that your mother in some ways can know better than you, when you're 4 years old, or I'm much older than 4, that really she pinned something that I think is really the central focus of this. I'll try to, a question I still can't wrap my head around, that's the focus of this talk, and really was the focus of my interview with Mac, because Mac went over the whole story of the Avery lab from the beginning through the discovery of the transforming principle of DNA. And that is, the question is, how did a pneumonia research lab that did the foundational work for these vaccines that millions of people are getting this season, and there's one you can see for baby. Pneumonia, of course, always, the people who were most susceptible to pneumonia were infants, young children, the elderly, and then there are people with risk factors. And I think this one here, the pneumococcal vaccine polyvalent pneumovax, that's given to them. And we'll see in a little bit that the absolute concept for this vaccine was developed in the Avery lab in 1929, 1930 by Wally Goebbels and Avery. There's just 2 of them on a paper. They had a paper series of 8 papers between, I believe, 1929 and 1936 on conjugate vaccines. I asked Vince a couple days ago, Vince, I don't understand why it took from 1929 to, I don't know, these started coming out in the early 2000s. And he gave the same answer that I did. I have no idea. So if there are any enterprising young science writers there, I think that would be an interesting topic. Why did it take so long? And what I want to do here is try to address this question of how did a pneumonia research lab find themselves in the position where they could discover that genes are made of DNA. And what we'll find is incredible parallels to the 2 stories of the research that led to this vaccine and the research that led to the identification of the transforming principle of DNA. And in fact, the connector between these 2 stories is Mac-McCarty. And the parallels between Mac taking this problem home to DNA and the way that the problem was taken home to lead to this, at least the concepts for these conjugate vaccines, are quite astounding. And I wish I could spend more time on this early phase of the research, but we do want to get to DNA, since that's what we're celebrating today. So I'm going to play. This is going to be the first. What we're going to do here is I have before I play the first little extracts, and there are going to be a series of 3 or 4 to start out the talk. Is we're going to follow Mac. He came to the Rockefeller Hospital over there, September 1941. And I'd like to initiate, if there are any outsiders to this story, you'll be initiated with Mac, because he didn't know anything about this story when he came. He had gone to medical school, I think started medical school at Johns Hopkins in 1933 when the last publication in the Avery lab on transformation had been published. So here's the first extract.
I knew about pneumococcal transformation from my bacteriology in Hopkins, but I didn't know they were working on it.
This is a picture that's in Mac's book, wonderful book, by the way, The Transforming Principle, which I believe was published in 1985. This is 1941. Avery in a kind of, he's obviously posing for the camera. We'll see a picture of him later on that I like much better. It kind of gives a sense, I think, a better sense of him.
And around the laboratory's office, and he would pad into the place, see one of the younger people, and sit down and chat with him and begin. They heard these tales too, these Red Seal records.
So I've always been fascinated by this. This is a picture of Red Seal records. It's a little before my time, but apparently this was like the gold standard for classical recordings of the 1920s, 1930s, when Avery's lab really started to reach its height. And the reason that people who knew Avery, whose nickname was Fess for professor, because even before he came to Rockefeller, he had a reputation for giving these informal discourses, especially to young people. He didn't like, apparently, to lecture to large groups. He didn't like to teach courses, but from his early days as a researcher, he loved giving discourses that, unfortunately, because I think the place had the money and somebody could have gone maybe to JDR Sr. and said, can we get this guy in who tells these wonderful stories to RCA, Victor, Red Seal Records, and record, but we don't have it, so we're going to have to try to reconstruct him. But we fortunately do have Mac as a guide telling us sort of his own Red Seal record. So apparently, they called it Red Seal Record because Avery would tell, there were several stories in the lab of different investigations, and he would start one of these stories about how we found this. And then we thought, well, what did that mean? And he'd played both sides. Did it mean this? But on the other hand, it could mean this. He'd reach in the air for a phrase as if he's trying to describe something. And it seemed this wonderful, spontaneous Shakespearean monologue or a form of oral literature. And it was because you could pass by the laboratory 3 days later and he'd be repeating the same story and he'd be reaching for that same phrase that he seemed to be reaching for. So Avery, if you're talking about Avery, you are talking about scientific storytelling. And I just bring this up briefly because we definitely want to do something with this 2-hour video, which we're just going to see 10 or 15 minutes, how you tell this story to a wider audience, because it is an undertold story. So this is a theme in the Avery lab, and maybe I'd love to talk to people afterwards about any ideas they have.
After we had talked about with his Red Seal record, I'd heard about the transformation, got brought up to date in the story and so forth, and I guess I asked enough questions to indicate some interest in the subject. So he, one day, still late September, he said, well, why didn't I sit in with him and run this test so I could just see what it was like? And that was about all it took.
And if you heard my voice after that interview, I said, and that's what you were doing for the next 5 years, are these tests on transformation. So as I said, he had several of these Red Seal records. Obviously, we're here to celebrate and to talk about the odyssey of transformation. But as I'll talk about in a little bit, there's an earlier epic story, which is the Iliad to the Odyssey of Transformation, which is the story that leads to that Prevnar 13, these conjugate vaccines that I showed at the beginning. So he's initiated into the mystery, and this is an image of, there's only one figure in this 1944 paper that we're celebrating. It's a plate, this picture right here. And we'll come back to images from this picture. Apparently, one of Avery's advice to a young researcher just starting to embark on his career was, the sort of ordinary researchers are always looking for the golden nuggets lying around the surface. But the special investigator digs a deep hole in one place. And he tries to find the right place, and if he hits a vein, he can make a tremendous advance. So we're kind of going to do that a little bit with this picture and the cells in this picture. This is transformation. These cells become these cells. We'll come back to this. These are cells that are completely harmless. If you injected these into a mouse, they'd be happy and alive, nothing would happen. You could inject, these is of course a colony of cells, a few of these cells into a mouse, they'll be dead within a day or two. And so what this will hear in a minute, Mac describe the appearance of these cells. This, what he will call shiny glistening, is a shield that the organism uses to protect itself against the immune system of its hosts, including, of course, human immune systems. And the purpose, of course, of vaccines is to initiate an immune response that defeats this shield, that defeats the defenses of these deadly pneumonia.
Just a glance, yeah. Matter of fact, that's really all we did for a long time. We just played out the culture, and if you could see whether you had transformation right away, because the colonies were the ones that were transformed that had capsules were several times as large as the others, as the picture shows, and glistened, they were shiny.
So that's what I think of them as, these glistening, shiny shields that of the foe, of the enemy that we're trying to understand and defeat. And of course, that was the aim of the Avery lab from its inception. This is a picture of Avery, this that I like. I think this is a north-facing window, so where Sloan Kettering would be here. He seems to actually be doing something, not just posing for the camera. And we're going to have a text slide coming up because, of course, the title of this talk is Discovering How the Genes Are Made of DNA. And if you look in the textbooks, of course, Avery-MacLeod-McCarty are somewhere between Mendel, Morgan, and Watson and Crick. They're in the history of genetics and ideas about the gene and the first connection between the gene and a chemical substance. But in fact, none of them were geneticists. They didn't set out to study, and we'll hear a little bit about this more later, they didn't set out to study transformation because they were interested in the secret of the gene. Not at all. In fact, it took them a little while to really understand the genetic dimensions of this problem. And I think that having done some history of science myself and gone to meetings and met a lot of interesting historians of science, it's easier to write about ideas than what scientists actually do in the labs all around here. And I'm going to show you a quote from Francois Jacob that will seem obvious to anybody who works in a laboratory. But in fact, if you read the history of science, it's a very rarely adopted perspective. So instead of talking about the ideas of the gene, ideas of genetics, idea of heredity, the alternative approach to studying the history of science and how investigations proceed over time, and in the Avery lab we're talking over decades, is to discover how objects become accessible to investigation, thus permitting new fields of science to be developed. These objects become the mysterious entities at the center of a domain which thought strives to explore, where it seeks to establish order and attempts to construct a world. And that's why I like that picture that I showed of Avery, because there's the outside world beyond his window, but he's clearly in his own internal world. And they're the objects of obviously the tools he's using, but the real object is this capsule. And this is something that they could see certainly in the 1910s, 1920s. The same glistening, shiny pneumococci colonies that you saw in the first picture from the 1944 paper. These are, I guess, Vince, these are 2 organisms? How many organisms? 4? 2? By the way, I am not a bacteriologist. I am not a biochemist. I do know a fair amount about what I was just disparaging, the history of ideas about genes, but that's why Vince is sitting right here. If I trip, he's going to correct me. So this was really the focus. This was the object that was the focus of investigation in the Avery lab. It's this that eventually led to the conceptual framework for the vaccine that I showed at the beginning of the talk. And it's this focus also that led them along the path of the transformation problem to DNA. And we're going to get a little glimpse of that. Mac's going to be our guide. So this is the figure with, just so you can, because I'm not going to get into textual analysis of the paper and the fact that Avery was much more cautious than either MacMcCarty or Colin MacLeod, and that perhaps the conclusions could have been sold more enthusiastically, as Mac did in a couple later papers in 45 and 46 when he wrote the discussion and introduction. In this paper, Avery wrote them. Just so you can see, it's a kind of, I mean, I don't know if this title would leap out at you. Studies on the chemical nature of the substance inducing transformation of pneumococcal types. So this is one type. This is actually type 2. We'll learn a little bit more about this. This is type 3. This is derived from type 2. Induction of transformation by, this is DNA fraction isolated from pneumonia. All right, we'll get into this later on. I just wanted to show it because we are celebrating tomorrow is the 75th anniversary of the publication of this paper. So I wanted to get into a kind of the prehistory of the stage of bacteriological research that Avery was engaged with. And just for the reasons of time, that didn't seem possible. But there's, just quickly, this led me to reread a book, The Microbe Hunters, which, how many people have read that book? Okay, well, a fair amount. I mean, and that was published in 1926. Paul de Kruijff was here at Rockefeller from 1920 to 1922. So at the time when Avery was here in the laboratory, he was fired by Flexner, the first director, because he contributed an anonymous article to a compendium volume called Civilization, which lambasted the medicine of the time as being gaga medicine and not scientific at all. Presumably, in contrast to the kind of scientific medicine that Avery and other people here were trying to do. But I guess Flexner thought that was bad public relations. And this is he talks about really the heroes, the founding heroes of bacteriology. I think his great hero, even more than Louis Pasteur, is Robert Koch. And I think if he had done a sequel from Microbe Hunters to Microbe Deep Investigators, I have a feeling, this is just my intuition, that his hero would have been Oswald Avery, who would have been the Robert Koch figure. And I was talking to Vince about this. I'm not sure that there was any greater bacteriologist of this period than Oswald Avery. So I'm going to, this is a picture. Here's Avery right here. I guess I should be using this. Does this work? Yes, okay. That's Avery, small man, physically. And so I think he's 37 at this time. He came over, he was working at the Hoagland Laboratories in a beautiful section of New York called Cobble Hill, attached to Long Island College Hospital, and I can speak to that because I've lived there basically since I was 10. He wrote an article, he was studying tuberculosis. He wrote an article which I think caught the attention of the first founding director of the Rockefeller Hospital, Rufus Cole, and he was hired to come over to what I think it was called the pneumonia service. In 1913, and he joined forces with somebody named Alphonse Daucher, who became a lifelong colleague and friend, and the two shared an apartment, I think until 1948 when Avery retired and moved down to Nashville to be with his brother. They were involved in something that seems, well, it is a century ago, in developing therapies for pneumonia, given the technology and the understanding they had at the time, and they were called serum therapies. And it involves the capsule, and it involves making antibodies to the capsule. And I have a little cheat sheet here to remind myself about the kinds of information that were available by the time Avery and Daucher were working together. And we'll come to this. Let me just play Mac for a little bit.
And they had recognized the fact that that there was a capsule surrounding the organism, and that there appeared to be a substance that was the capsule was composed of this substance that was responsible for the type specificity, so that Daucher and Avery began trying to determine the nature of the capsular substance.
So let's go back to that Indian ink image. and what was known when they started working together, and as they made their first big discovery, and Avery's first big discovery in his career between 1916 and 1917. So I mentioned Robert Koch before, the discoverer who really showed how you can say a single microbe causes a single disease. Koch's postulates of disease with anthrax, then went on to discover the bacteria responsible for tuberculosis. An institute was created in his name, as was one for Pasteur. And of course, those were major influences on the creation of Rockefeller Institute for Medical Research. And somebody was working at the Koch Institute between 1909 and 1910, Fred Neufeld, discovered this pneumococcus was thought had been linked to pneumonia in the 1880s by several researchers, but it was thought to be one type, maybe what they call species. And Neufeld found that, in fact, there are at least 3 types. And the way he found this was he was studying pneumonia patients, human patients, isolated the pneumococci from different patients, and injected them into mice who had been the pneumonia had also been, the specific types had been injected into horse and rabbits, who of course make antibodies because they're under attack. And what he found was you could type this pneumonia into 3 types based on which antisera protected which type. That wasn't as clearly stated as... in other words, already by 1910, there's a recognition that there's something on this capsule that's chemically different between the 3 types, and the way you can tell the difference is 3 different types of antibodies. And that is why, as Max said in a clip that I did not include, you can get pneumonia from type 1, and if you recover from it, don't die, you will never become infected with that again, but you can get pneumonia from type 2. And if you get type 2 and recover from that, you can get it from type 3. So antibodies are type-specific. That's the different serotypes of pneumonia bacteria that were defined by Neufeld. He also found that there seemed to be something, the antibody was binding to something in the capsule, or at least at the outside of the organism, because if you had a test tube full of a particular type, let's say type 1 pneumococcus, and you had antisera, type 1 antisera, and you put in the test tube, they'd agglutinate. They group together the cells. The other thing that Neufeld discovered is the Quellung phenomenon, the swelling phenomena. So if you look under the microscope with this India ink, and you have type 1 in the presence of type 1 antisera antibodies, the capsule will swell. So it seems to be that the antibodies are binding to something in the capsule. I'll go to the next.
That was the implication. Obviously, without knowing what it was binding to chemically, all they knew was the structure. It was obviously combining somehow with something in the capsule.
So Daucher and Avery together, and this is a great story, and I'm going to have to, unfortunately, really abbreviate it. Daucher, I think, makes the first discovery that if you wash this pneumococcus with salt, just a salt solution, something washes off that was called, washes off into solution that precipitates with specific antisera. They had the idea, so it's something, it seems as if the surface of the bacteria is coming off, and it's in this soluble substance, which they call soluble specific substance, and precipitating with specific antibody. They had the idea, Avery and Daucher, that they found it in the bloodstream of patients, and then they said, well, maybe it's going into the urine too, and in fact it was. And you could detect this soluble substance in urine. You could type what type of pneumococcus a patient was suffering from just through a urine test. So this was the big breakthrough, Avery's first big breakthrough, because this place was involved. There was a heavy effort to make it specific antisera through injecting horses to treat pneumonia, and they had a pretty much success with type 1, but not with type 2 or type 3. So they were trying to improve this. And as it now turns out, the reason that the horse anti-sera for type 2 and type 3 did not work is that antibodies, Vince gave me a little tutorial the other day, have two sides. One binds the antigen, in this case the capsule, but the other side, studied here by Jeff Ravitch, and of course many others, is the effector side that signals to white blood cells to phagocytose, to swallow the organism to which antibodies are bound. So the horse anti-sera for type 2 and type 3 did not have that connection to the white blood cells that actually cure disease, protect you against pneumococci. So I think the interesting thing, at least for me, is somebody has done, and Jeff Friedman in the audience, I think, was just at the Rockefeller Archives looking at these. If you're trying to track a Rockefeller researcher, what they do over time, they had to do these quarterly reports to the Rockefeller scientific directors, and you can really track the progress of a lab. And it's very clear that from 1916 to 1917, even though there's this specific soluble substance, the orientation of Avery's lab or unit within a larger pneumonia service was very clinically oriented. And in fact, he was the whole lab was involved in World War I in creating anti-sera for soldiers. And somehow between World War I and 1922, Avery takes an increasingly chemical approach to his investigation of pneumococcal pathogenesis and immunity. And I wanted to play this music because I've always been kind of haunted by it. That René Dubos said Avery was a very social guy, but when he'd be alone in the lab, and I heard this from other people, from Roland Hotchkiss, who was also in the lab, he was prone to move slowly from one object to another, gently whistling to himself this from Tristan and Isolde. And I do find this very melancholy. Avery's own brother, there's a wonderful picture of him with his older brother. Both think he died of tuberculosis. His mother died of pneumonia. So these were not abstract problems to Avery. And what happens to a researcher when obviously he was being promoted within Rockefeller, so perhaps he felt more confident about planning long-term research programs. But in any case, even though they had discovered this specific soluble substance in 1916, 1917, they made no headway to identifying what it was chemically. And Avery obviously thought that this was an important thing to do.
What Avery did, and his idea was that it would be important to know the nature of this material. And he had this product which they extracted from the cells that was reactive with the antisera. It wasn't identified chemically. It wasn't a pure product. It was this brownish material in a tube, but it was very reactive with the antisera. And he started on Heidelberger, who was a biochemist.
So Heidelberger was working for Van Slyke, Donald Van Slyke, who was the clinical chemist at the Rockefeller Hospital, and he was crystallizing hemoglobin. And Avery, with a version of the Red Seal records, is trying to seduce him into working on his problem.
And he wanted to get Heidelberger interested in this. And he'd keep showing him the tube and saying that, when are you going to get around to working on this?
This is another little passage of music from that Tristan and Isolde that's a little more seductive, tantalizing, and in my mind at least, this is playing while Avery, and we have Heidelberger's word, that Avery would go over to wherever Heidelberger was working with these tube and wave it at him. When are you going to come work on this, Michael? The whole secret of bacterial specificity is in this vial. Does this happen in the university today, that this music plays when they're trying to get you to come to your... So this is a Heidelberger in 1977 wrote a memoir of his time at Rockefeller, and he is recruited in the problem. Type 2 is the first serotype that they try to identify the chemical identity of this capsule, of this specific soluble substance. Precipitation with alcohol and centrifugation, they found this gummy middle layer. And the more, I should say that they expected that this capsule would be a protein, because an antigen at that time was thought to be, all antigens were thought to be proteinaceous in nature. So the fact that it didn't have nitrogen meant it was not a protein. It surprised us. All immunologically active substances were supposed to be protein. Finally, when it was nitrogen-free, Avery said, could it be a carbohydrate? So, of course, this is the first parallel between the soluble specific substance story and the DNA story, where at the time that the Avery lab was pursuing the chemical identity of the transforming principle, everybody thought genes were made of proteins. In this case, they thought the capsule would have to be protein, but no, it was a carbohydrate. Now then they went on the type 3, soluble specific substance purification. Now, type 3 is the model that is used in the transformation experiment. That is the shining, glistening cells that Mac was talking about in one of the clips at the beginning. As soon as we started to purify our first batch of the soluble specific substance of type 3, I knew it would be different chemically from type 2, as instead of type 2's gummy layer, it formed dense fibers when precipitated by alcohol. If this was a detective story, we'd shine a light on this little clue and a little red herring, because, note, precipitated by alcohol forms dense fibers.
You know, the idea was there could be a little bit of protein in this, and since proteins were usually the typical antigens, that was all you were seeing. Avery got the idea that if you could find an enzyme that would specifically attack the polysaccharide, and it would destroy this activity, why then you would have a way of eliminating that skepticism.
So Mac has a wonderful description of a famous anecdote when Avery meets René Dubose in the famous lunchroom, which I believe was downstairs. René Dubose was not a physician, not a medical researcher. He was a soil microbiologist, and Avery told him about his idea to find an enzyme. And they had tried all kinds of different animal and plant enzymes to attack the polysaccharide from the pneumococcal capsule, and none of them had worked. And Dubose was convinced, since these soil microbes have to break down everything that's coming down into them, including cellulose, which itself is a polysaccharide, that he'd be able to find one. And he was able to find one, and that's this S3 enzyme that really proved that the capsule was a polysaccharide, and the polysaccharide, of course, would precipitate with the type-specific antibody if you treated the polysaccharide with the S3 enzyme. You no longer had that precipitation. So, specific enzyme destroys specific activity. This is somebody who joined the lab.
Wally Goebel joined the lab as these things had gotten fairly well established, and did some very nice work with respect to getting synthetic sugars for polysaccharides or oligosaccharides, a few sugars attached correctly that would have specificity that you could, matter of fact, you could make antibodies to those synthetic ones that would protect mice against the infection of that type.
So again, this is the foundation of the conjugate vaccines that we saw are now all around us being given. This is the first of this 9-paper series published in 1929, 90 years ago, that lays the foundation. Transition to Odyssey of Transformation. So While Goebbels and Avery are doing this work, in 1928, there's a person in the lab, Martin Dawson, who's studying these R cells. And if you remember, that's on the left side of that figure 1 from 1944. And what R cells are they're a laboratory-derived strain that comes from getting an encapsulated cell and growing it in the presence of specific antisera. And what we now know is you're really conducting a Darwinian experiment, and in order for those encapsulated cells to survive against the antibody, they lose their capsule. So these do not have a capsule. By losing the capsule, they also lose virulence. But this is an interesting problem, because if you're interested in the capsule, how do they lose the capsule? We now call these cells mutants. They're losing one or more genes, or having a partial loss of function or total loss of function of genes that are involved in capsular synthesis. But, as Max says in the next there was not a real, the idea that bacteria even had genes was not at all accepted in the 1930s.
Bacteriologists knew that they could get different variations, and they talked a lot about bacterial variation, and they just didn't use the same terms as geneticists. They didn't call them mutants when they got something, but they had a lot of variants. And it was simply because bacteriology hadn't gotten in the main line of biology yet.
While Dawson is doing these experiments with the R cells, word of an experiment from London, Fred Griffith, the famous transformation experiment that involves these R cells. And here's the basics of the experiment. And I'm sorry, telling these stories with these type 2R, type 3S, it can get a little difficult to follow, but I'll try to do it as simply as possible. So the R cells, by definition, are non-virulent, non-encapsulated. If you inject them into a mouse, type 2, the mouse lives. Heat-killed type 2S cells, which, if they were alive, would be virulent, but because they're dead, are not virulent, again, the mouse lives. But if you inject them together, the mouse dies. And from the body, you recover virulent type 2, living type, this is a mistake, type 3S, that's a mistake, type 3S pneumococci. So the conclusion is that somehow type 2 cells have been transformed to type 3 cells, a property inherited by all the cell's descendants. So this is the transformation phenomena that the Avery lab would then be studying for almost 20 years. This is a conceptual animation of transformation done by Joe Alexander at IT. And just to maybe get an idea how strange this phenomenon was and what the meaning of it was. And in fact, the Avery was interested in this possibly because of its medical implications, because, as Max says in another part of the interview, because capsule was associated with virulence, understanding capsule formation could lead perhaps to a therapy that could prevent this process. Okay, so now we get into the real odyssey, which is you have this process of transformation from type 2R to type 3S. How is this happening? They called this the transforming principle. What is the transforming principle? Now, of course, they thought it was some kind of biochemical substance. And in order to, and Mac, in some of the things he's written, has shown some of his laboratory notebooks of his experiments. And they do look sort of like a bank ledger and an accountant, because you're tracking your assay of activity versus amount of material you're using. And Arthur Kornberg, of course, one of the greatest biochemists of the 20th century, who founded the Stanford Biochemistry Department, or at least the founding chairman, which I think some people, like Mike Young, learned how to work with DNA. In his book, For the Love of Enzymes, says he didn't think of the work that he did purifying in terms of being a banker and an accountant. Rather, the process of purification often seemed like the ascent of an uncharted mountain. The logistics resembled supplying successively higher base camps. Protein fatalities and confusing contaminants resembled the adventure of unexpected storms and hardships. Gratifying views along the way fed the anticipation of what would be seen from the top. So what we're going to do is we are going to visualize the 10-year odyssey of the Avery lab purifying the transforming substance as DNA in terms of establishing these base camps. And we'll go quickly over some of these steps, but I like to, so time will be visualized as space, and we'll have names and dates here. The first step, so Dawson repeated the Griffith experiment in the Avery lab, went to Columbia. Dawson and Sia found that they could take the transformation process out of the mouse's body, but they still required some intact cells. Lionel Alloway in Avery's lab was able to remove the intact cells and cell debris. So this is apparently, according to Mack, a experiment suggested by Avery that he pass the crude transforming extract through what's called a Berkefeld filter, and that was the Berkefeld filter was the filter that was originally used to define viruses as distinct from bacteria, way before the days of electron microscopes. Viruses were originally called filterable agents. So like viruses, whatever the transforming principle was could pass through this filter, and the transforming principle would lie somewhere in the soluble substances that pass through the filter. So this is the first base camp at the base of this mountain that makes this into a system that one can try to purify what that substance is. Colin MacLeod, who of course is on the 1944 paper, came to the lab in 1934. Avery was suffering from Graves' disease and had stopped doing experiments, but still was around the lab at least occasionally. And Mack thinks that, just as he did later, he received some of the Red Seal records on the transformation story and was seduced into the problem of transformation. He regenerated that line of investigation within the lab, and he made the second base camp.
He went through this process of passage, and it was 36 passages in the presence of antiserum. He got this strain called R36, which was actually used from then on for all the experiments, and still his descendants from that are being used.
Okay, so here we have base camp number 2, Colin MacLeod, 1934. These are the exact cells. These are R36 cells that were in the left side of the panel, the 1944 figure. And he developed this is the model system that's used for all the subsequent experiments. However, and I find this interesting, that they did not immediately interpret the transformation process as a genetic process. Instead, they thought of it in what we now call as a case of enzyme induction, that all R cells possess a potential but inactive system of enzymes capable of synthesizing any of the type-specific polysaccharides, a particular one being produced being determined by the specificity of the activating stimulus. So in modern-day terms, the transforming principle would not be a gene that's being transferred from the dead type 3 cells to the living type 2R cells. It would be a trigger that would trigger an innate potential to make any kind of capsule that the R cells would possess.
And did not, at that point, focus on, at least in the write-up, the possibility that it was some kind of a genetic change. The point at which the thinking began to change is something that's never been defined well. I feel quite confident it was probably already germinating even at the time of that 1935. It was very hard to work on that problem for very long without getting, when it started myself, it just, you couldn't believe that, first place, there were so many different pneumococcal types, you could hardly believe that wide you could take that same type 2 R36 strain and I'd myself ran it through to type 14 for a reason, to type 17, a type 26, you could do any of them practically. So that seemed terribly unlikely, plus the fact that it meant that that inducing factor, whatever it was, had to be replicated in the cells, because you could take the transformed cells and get more transforming activity out of them, so that it was being replicated. That, this was, as you started thinking about it in these terms, it was very hard to believe it was not genetic.
So the next stage is MacLeod uses a chemical process to remove protein from the transforming extract, and this corroborated previous work that had used proteases, protein-splitting enzymes like trypsin. And I should say that this is one of the great things about this story, both parts of the story, is how much converges on the Avery Labs investigation of the chemical nature, both of the polysaccharide capsule and of the transforming principle, because all these enzymes that were used, a lot of these enzymes were actually purified and crystallized by Rockefeller scientists, usually at the Princeton laboratories, which then existed, Northrop and Kunitz, and Northrop would eventually win a Nobel Prize for this kind of work. So it did not seem, just as the capsule, the soluble specific substance, was not protein, it did not seem as if the transforming principle, the transforming substance, was a protein, or if it was, it had to be resistant to this chemical treatment and these proteases. The next stage is, again, RNase is a enzyme that depolymerizes RNA. The nucleic was the only nucleic at that time that was recognized to be in pneumococci. So it's kind of hard to believe, but nobody had ever identified DNA as even existing in pneumococci before these experiments, before the Avery lab experiments. So they use RNase, and the other thing I want to mention is that RNase was an enzyme that was purified, crystallized by Kunitz, again at the Princeton labs. It formed the research topic of several very distinguished biochemists at Rockefeller, Stein and Moore, and Bruce Merrifield, Nobel Prizes. So it's a storied history and intersection of research paths within this one experiment. This is from Mac's book, and Mac, one of the wonderful things about Mac's book, The Transforming Principles, he went over all the laboratory notebooks dating back as they then existed and tried to trace the story. So this is an indication of how far out it seemed that DNA could be the transforming principle. In January 28, 1941, MacLeod writes in lab notebook, "Thus it would appear as though these transforming extracts may contain a little DNA in addition to the large amount of ribonucleic acid present." So it was a surprise, and as Mac, reading that notebook entry, sees that Colin MacLeod's initial skepticism. So I'm going to skip over this. Avery had not been conducting experiments, but he is revived by MacLeod's progress. He is doing experiments with MacLeod. Mac has a nice passage about this in his book, about how they had the transforming extract, and Avery took one half of it and did it his new way, and MacLeod did it the old way, and Avery's way was much better, and that's the method they used for all the subsequent experiments.
Ran into the same old problems of variable extracts. Sometimes they worked, and sometimes they didn't. And we're struggling along with that and trying it. Then in January of 41 was the first recognition that DNA was present in their extracts, which shows you how long that took.
So this is what the previous slide discussed, that it was in these ribonuclease experiments that the first evidence that DNA was in there. Now, let's play this.
It was thought to be the polysaccharide. Because it had been found earlier on that the polysaccharide, when you precipitate it with alcohol would give these long fibrous strands. And so that this was interpreted as being the what they call the specific soluble substance of the polysaccharide that was coming out that way. And I think this is the confusion that came about then in June before MacLeod left, was the fact that there was that apparent loss of activity if you lost the polysaccharide.
So I should have set this up a little better. During the purification process, as they purified with alcohol, a fibrous substance came out of solution. And as you remember, Heidelberg described, they're working with type 3, that if you precipitate in alcohol type 3, the specific soluble substance forms these dense fibers. So I'll give you a little spoiler. In fact, it was not polysaccharide, it was DNA, but they misinterpreted it because it looked like polysaccharide. Heraclitus used to say nature likes to hide, and it also can trick you. There's MacLeod-McCarty in about 1940. He came to the lab in 1941, and the first thing he needs to do is to clear up this confusion about polysaccharide, because I think, as he says in the next clip, not only was it thought that these fibers could be polysaccharide, but that somehow the transforming principle required may not be polysaccharide, but required polysaccharide is either a template or a primer to make more of this capsular substance.
I knew about pneumococcal transformation from my bacteriology in Hopkins, but I didn't know they were working on it.
Really? Until you came in September, you weren't.
That's right.
And of course, there hadn't been any publication since
that's right.
since Alloway in '32.
Yeah.
Did you know about Colin MacLeod's work at that point?
No. This was all something that hadn't surfaced.
So a point I want to make, and it was setting it up, that when Mac first came to the lab in September 1941, he heard several of these Red Seal records, which were the classic stories of investigation in the Avery lab. And of course, the ongoing odyssey was transformation, but we also had the Iliad of the soluble specific substance. And in that story was the use of the DuBose enzyme to prove that the soluble specific substance was in fact a polysaccharide. So Mac now goes back to use that enzyme to show that the fibrous substance that seems to be involved in transformation is not in fact a soluble specific substance.
And that first step that I got involved in was getting the DuBose enzyme cranked up to where we had enough of it. DuBose had some that had been stocked away that he provided me with, and then I also began to make it because we had the organism, and just growing it in the presence of the type 3 polysaccharide, you could generate more enzyme, which I did. But I just did these experiments, running into all the problems that one did with the system and all. But finally, by December, I had a clean experiment where I had eliminated the polysaccharide with the enzyme from the extract to where it was no longer detectable, and it was turned out the test on the material afterwards was the most potent. That's one that went out to 1 to 10,000. So you had the feeling that you knew for sure that the polysaccharide had nothing to do with it. And it's out of materials of that kind that we suddenly became aware that there was still fibrous precipitate.
Okay, so we're at base camp 6. Mac using the DuBose S3 enzyme to remove polysaccharide, and the confusion about the role of polysaccharide, and getting a transforming activity that's more potent than any previous extract.
And finding out that it was the active material was very large, and would spin down in the ultracentrifuge and could actually be spun down to where it was, you could just pour off the supernatant, and this material was still a sort of a sticky precipitate down at the bottom. But that's where all the DNA was again, you see, and that's where all the activity was.
Okay, base camp number 7. We're getting close. Ultracentrifuge spins down DNA and transforming activity in parallel. In March 30th, Mack did an experiment. The nature of the material in transforming extracts giving stringy precipitate and alcohol. Results strengthen suspicions DNA is a transforming principle. Ultracentrifuge experiments provide independent evidence for the same suspicion. So at this time, as they're becoming more and more convinced that DNA seems to be the transforming substance, they have to confront the skepticism that DNA could possibly be the genetic material. DNA was thought to be a stupid substance in which the 4 bases were repeated in the same order over and over and over again. They couldn't have specificity. The DNA from one species would be the same as the DNA from another species. Mack went, at this time, it was probably in 1943, because Max Bergman was a very distinguished protein biochemist. And he went to talk to him about whether, in fact, there could be, whether nucleic acid was all the same or, in fact, could have many different kinds of specificity.
In the light of present knowledge, the statement that all nucleic acids are the same regardless of the source from which they are derived is nonsense. If they are large polymeric compounds, there's an endless number of possible combinations, all of which would possess the same elementary composition, but would differ in chemical structure nonetheless. Nucleic acids hold too prominent a place in biology to be completely nonspecific substances. The lack of evidence of any specificity associated with nucleic acids is only due to the fact that they have not been investigated sufficiently.
All right, now we get to the top. I'm telescoping time a little bit. In the paper, the 1944 paper, 75th anniversary tomorrow, and beginning in 1942, Mack started using a crude enzyme fraction that had depolymerizing activity on DNA. And that had been studied by other people. The one advance that he made was to get, not just to treat the pneumococcal DNA and show that that was depolymerized, and to show that that same enzyme fraction destroyed selectively transforming activity, but to get calf thymus DNA from somebody named Alfred Mirsky and to show that it had the same depolymerizing activity on what that was the standard source of DNA at the time, calf thymus DNA. At the time of the paper was published, February 1st, 1944, Mack had not gotten beyond the crude fraction, but he pursued that problem, and at the end of that year, he gave a Friday lecture on the purification of DNAs. And I think that this is something, in fact, in Kortenberg's book, For the Love of Enzymes, talks about how this is unrecognized, both the enzyme and the discoverer. This was later purified by Moses Kunitz in 1948. And in fact, if I'm not mistaken, somebody could correct me, this is the first enzyme that was ever purified that acts on DNA. And it was the first tool that biologists had to play around with DNA. And of course, many other tools for playing around with DNA were subsequently developed. And it turned out, we've been talking about bacteria and immunology, of course, that bacteria have their own immune system, something like an innate immune system. It was a study of that, out of which restriction enzymes came. And they have also something like an adaptive immune system. And of course, it's studied out of that the CRISPR-Cas9 system came, which Luciano Marafini here, of course, showed that people thought that was acting on RNA. In fact, it's acting on specific sequences of DNA. So I think that this is something that it is, if the Avery lab, if the paper tomorrow is perhaps, on the one hand, recognized as an incredibly historic achievement, but is an undertold story, Mack's purification of the DNase is even more undertold. And I want to make the connection also to the Iliad of the soluble specific substance, because Mack learned the lesson of those Red Seal records on the soluble specific substance, I think, better than anybody. Because with the DNase is the parallel to the Bose enzyme. You get a specific enzyme, destroys a specific substance, destroys a specific activity, and that really was for most people, the proof that DNA was the transforming principle, because Mack, when the paper that was published 75 years ago tomorrow was the first in a series of 3 papers that Mack wrote with Avery. And in one of those later papers, he writes a discussion. Avery had said in 1944, if this is shown beyond a reasonable doubt. Mack, in one of these later papers, after he's purified DNA, says this has been shown beyond a reasonable doubt, essentially. I have to show this because this is a famous letter that was written by Avery to his brother, Roy, who was a bacteriologist at Vanderbilt. It's one of the only glimpses we have of a sort of more private side of Avery. And he talks about the whole transforming process, the Odyssey. The crude extract, what it has, try to find in this complex mixture the active principle, try to isolate and chemically identify the particular substance that will induce transformation. Some job full of heartaches and heartbreaks. And this is one of my favorite passages in the literature of science, and I'm going to play another little piece from that Tristan. When you put alcohol in, there separates out a fibrous substance which, on stirring the mixture, wraps itself around the glass rod like thread on a spool. And I love this spiraling music, because if you're ever going to have spiraling music, it should be for DNA. So here's a summary slide of the ascent up Mount Purification. And you can read it. We went through all the steps. It seemed pretty fast when we were going over it, but look at the time, 11 years. Heartache. So this is the second part of Arthur Kornberg's mountain climbing quote. As you're going, you're looking up at the top. What happens when you get there? The ultimate reward of a pure enzyme was tantamount to the unobstructed and commanding view from a summit. Beyond the grand vista and thrill of being there first, there was no need for descent, but rather the prospect of even more inviting mountains, each with the promise of even grander views. Now, of course, there are many wonderful enzymes that have been purified and will be purified, but I will say I do not think that any chemical substance ever purified will equal the vistas afforded by DNA. It's changed. I will not try to wax poetic about what DNA is and how there are many people in this university and in this audience, I'm sure they could do a better job. But obviously it's changed every area of biology, every part of our understanding of life, of evolution. And this just one little glimpse. This is actually, of course, the advances in DNA sequencing technology have been incredible over the last 15 years. So this is a 2006 paper out of the Sanger Institute. They sequence all 94, because it's not just 3, there are 94 serotypes of Streptococcus pneumoniae. These are the 3 types that were studied by Avery. Here are the genes for the biosynthetic, for the enzymes that are involved in the biosynthetic pathway for capsules. So here's makes type 3 capsule, type 2 capsule, type 1 capsule. This is what you can see today on your computers.
At that point, there was nothing like that. You see, also nothing that the geneticists had a handle on so that they could test for what the nature of the gene was. And really, it wasn't clear, I suppose, at the beginning that that's what transformation was, but it turned out to be, I think, perhaps the only procedure at that point that had ever been found that would allow you to test for the nature of what was a genetic transfer.
And Vince alluded to this in his introduction. some people got the message of the 1944 paper, but it took time. And I asked Mac, were there geneticists that did get it?
And I met him at an affair. Actually, it was Mirsky that had arranged it. And I was there, and he said, I don't know whether you fellows know it or not, but you're working on genetics.
That's the end of, we have, don't you like those movies where they have bonus video clips when the credits are rolling? We have a couple bonus video clips that Olga found. And first, I'd just like to thank some people that helped prepare the talk. First of all, I want to thank Olga for asking me to do this, and Matt for hosting me. I want to thank Jeff Hannafeld for doing great work with the video, and Todd. I want to thank somebody, a graphic designer in the development office called Kathy Vanchieri, for her exquisite visual taste and for finding that picture of Mount McKinley, which we used. And I should thank Torsten Wiesel for hiring me and giving me a lot of freedom so that I could do fantastic stuff. But the person I most want to thank, and I'm most grateful to, I want to bring in a special guest via video to help me pay tribute to him. It was really an honor to give this talk and to get to know, this is Oswald Avery, to get to know in an angel's outfit, to get to know Mac a little bit and to do this interview. And I think Dr. Avery would join us in paying tribute to Mac for bringing the odyssey of transformation home to DNA, the seed and root of life. So to Mac, thank you, Mac. And thank you all for coming to this 75th anniversary celebration.